Vanakkam. Welcome to this course on classics in neuroscience. In the last set of videos, we have been discussing about neurological foundations of neuroimaging or brain imaging. In this video, we will look at the spreading depression of Leo and migraine. This is a topic that is slightly different from the rest of the topics which are on imaging. This is a topic on migraine and spreading depression. But since we are discussing cerebral blood flow and we are discussing the developments of the 1950s, it would be incomplete to not describe the contributions of Leo and spreading depression. So, Leo was born in 1914 in Rio de Janeiro in Brazil to, I think, well-to-do or affluent parents, and he entered medical school, but somehow due to some illness he could not finish medical studies. So, he dropped out of the med school, and in 1940 he decided to travel to the United States because he was generally well-to-do and he just wanted to explore and study the United States. So, he went to Harvard and he was accepted into the physiology program at Harvard Medical School, where with Hallowell Davis and Arturo Rosenbluth he started studying, and for his degree project he investigated the topic of cortical epilepsy. That is, when you expose the cortical surface of an animal, cerebral cortical surface, and you electrically stimulate the cerebral cortex of that animal, there is a spread of activity throughout the cortex. This is what was the project that was pursued by the undergraduate student, that is Leo. What Leo found, to his shock or utter surprise, was that in response to an electrical shock, the cortical response was a spread of depression in surrounding region that has an ongoing electrical wave activity, not a spread of excitation. So, he found that in the rabbit cortex there was significant and enduring reduction in spontaneous electrical activity. There was a suppression of electrical activity that was then followed by the tonic-clonic activity that is similar to epileptic seizures, but first there was the suppression of electrical activity in response to an electrical shock. This suppression of electrical activity is called, or came to be called, as the spreading depression of Leo. And Leo suggested that this spread of tonic-clonic responses and the depression that preceded it were essentially having the same cortical elements, that is, both of these involve same cortical structures and cortical elements. The depression followed by the tonic-clonic response, which is reminiscent of seizures, epileptic seizures. Both of these arise from same cortical elements. Also, Leo showed that in PL blood vessel there was vasodilation and increased blood flow in association with depression or spreading depression. This is perhaps the first demonstration of an increase in blood flow when there was no excitement of neurons. Depression essentially means a reduction in the excitatory levels of the neurons, but still there was an increase in blood flow. This was perhaps the first demonstration of this phenomenon. So, Leo and Morrison showed a causal link between the slow spread of depression and the drifting movement of scotomas in migraine. But there was a lot of skepticism surrounding this research and these results. Many of these came from Leo's own advisors or mentors, PhD mentors. Remember, these results, revolutionary as they may be, came from the work of a graduate student, a PhD student. So, his own PhD mentors were kind of skeptical about these results, noting that nothing resembles a new phenomenon as much as an artifact. By the way, this is a good attitude for mentors and students alike to have. Whenever you think you have discovered something new, please check if it is an artifact, because more likely it is an artifact. But of course, if you are able to reproduce this many times under more controlled environment, maybe you have found some new phenomenon. But first verify whether it is an artifact or a new phenomenon. Although there was this initial skepticism among researchers, in particular mentors of Leo, later a lot of researchers who are working on epilepsy and related fields were able to reproduce these results. Also, the observations by Martins Ferreira in this field, highlighting the reproducibility of spreading depression in the exposed retina of a frog, where a dark spot growing into a dark disc and continues to grow at a slow but steady rate, finally engulfing the entire retina with sharp borders, was able to demonstrate that this spreading depression. Of course, there were others who corroborated this research. For example, Peter Milner's work in 1958 corroborated Leo's spreading depression with Carl Lashley's description of the rate of scotoma movement. Carl Lashley had migraine, and he essentially described the rate at which his migraine proceeded, scotoma. Yes. So, he was able to, Peter Milner was able to relate Carl Lashley's rate of scotoma with the Leo's spreading depression. Possibly spreading depression is also related to transient global amnesia and cerebral vascular disorders. Now, these things or these topics such as spreading depression are studied using functional magnetic resonance imaging, functional neuroimaging, or diffusion weighted functional magnetic resonance imaging and magnetoencephalography to track spreading depression in humans. With this, we come to the end of this video. In this video, we looked at the seminal contributions of Leo to the study of spreading depression and migraine, and how works of great significance, great importance can come from the sincere, dedicated efforts of PhD students. So, never give up on yourself and stand up to your own results when you find them. But of course, maintain this healthy skepticism that nothing resembles a new phenomenon as much as an artifact, but do try to reproduce your results. If you do get the reproducible results, then maybe you are onto something. With this, we come to the end of this video. Thank you for your attention.